Okay, so one of the perks of this job, being president of the American Society of Plant Biology, is that you get to organize a symposium. And it can be whatever you want it to be. And I wanted it to be something that I was very passionate about. And it could have been my research, because I get very passionate about that as well. But I wanted to go a little broader and look at something that I think as plant biologists and really as human beings on this planet, we should be quite concerned about. So I say, can I have the first slide? That genetic diversity can be a beautiful thing. And so I wanted to focus on genetic diversity because I think it is the key really to our future. I would say that we as plant biologists may understand that. We may not embrace it fully, but we understand it. I'm not sure that the average person walking around on the mall does understand it, but I think as plant biologists, it's really sort of our role to help people understand how important that is. And I think the first time that I was really alerted to this was something that came out actually in the National Geographic. This was a couple years ago. And I saw this beautiful image, and it basically said, well, you know, here are all the varieties of these various different crops that we had back a while ago. And that looked really interesting. And then it said, but look what's happened now. Look at the genetic diversity that's out there now. And that was really shocking to me. Because, and I think that that's our resource. That's what we can build on in the future. And the more that that is diminished, I think the worse off we are as human beings. So what I just tried to do was to try to bring together some individuals that can help us understand that in a better way. And I'm really lucky to have 4 really excellent speakers. So we're going to hear from 4 individuals, Carey Fowler, Phil Pardee, Jonathan Lynch, and James Dale. And they're each going to take a different view of genetic diversity. So our first speaker is Carey Fowler. And I first became aware of him by reading his book on natural selection. I thought, this guy's really interesting. And then when I knew he was interested in diversity, so when I was trying to get this talk together, I went on Google and I said, well, you know, what can I find? And I found his TED Talk, and I watched his TED Talk, and you should do that sometime. It's really interesting, and he was just telling us an interesting story about what it's like to give a TED Talk. But I watched that and I said, I want him. I want him to come and talk, but I didn't think there was any possibility that I would be able to do that. Julia said, ask him a year ahead of time and maybe he can make room. And so he did. And so I'm really very pleased that Carey was able to come and join us. He's been involved in germplasm and germplasm issues for a long time, and was one of the initiators of the Svalbard Germplasm Center in Iceland, and I'm hoping he's going to touch on that today. So, Carey, it's our pleasure.
I think you're the only one that's read on natural selection.
Oh, no.
Thank you very much. Since the dawn of agriculture in the Neolithic era, when the population of the world was about the size of Colorado today, the single most important factor in increasing food production has been simply to increase the amount of land devoted to growing crops. Increasing production simply meant cutting down trees and using more land. But in the 1980s, something quite remarkable and historic took place. For the first time in history, this factor of increasing food production through increasing cropland took second place, backseat to increasing yield. Now, historically, about 50% of the increase in yield has come from genetics. And if you consider the fact that we are going to have and do have land constraints, water and nutrient constraints, and not to mention the need of plants to adapt to climate change, then obviously genetics is going to remain important in the future to food security. So it's in this context in which the fate and the health of gene banks should be a concern to all of us. Now, the pedigree of modern gene banks goes back a long way. You have Egyptians and Sumerians thousands of years ago, literally, organizing the first plant collecting expeditions. In the modern colonial era, you had colonial powers that established networks of botanic gardens where they acquired, tested, developed, and disseminated economically important plants. And in the United States, we also had plant collecting expeditions and large programs of introducing seeds and planting material from abroad and distributing those to farmers. More on that in a minute. But it really wasn't until the commercialization of agriculture and the rediscovery of Mendel's laws of heredity in 1900 that interest turned in earnest to not just to the diversity between species, but to the diversity within species. The modern era of gene banks really began with the Vavilov collections assembled by Nikolai Vavilov in the Soviet Union beginning in the late teens and early 20s. The first cold storage, long-term storage for seeds, gene banks, took place in this country in Fort Collins, Colorado, starting in 1958. Well, by the early 70s, with the Green Revolution underway, scientists became concerned about the loss of genetic diversity, about genetic erosion in the fields. And the leading figures at the time, there weren't too many, the grand old men and the grand old woman, there was one, Erna Bennett, got together, and they thought that at that time that the world needed probably about 6 to 8 international gene banks that could provide long-term storage. By the mid-'80s, we had 72 countries with gene banks. There were 52 long storage units and 2.5 million samples. In the 1990s, by mid-'90s, you had 137 countries with gene banks. There were about 76 gene banks offering long-term storage, 6.2 million accessions. Fast forward to today, I don't know how many long-term storage units there are. There are about 1,700 gene banks, and there are about 7 million accessions being stored in these gene banks. So obviously the grand old men and the grand old woman back in the beginning were off by some factor, at least in terms of what was going to happen. They might have been correct, however, in terms of what needed to happen. So these are very impressive numbers. And I don't want to belittle the contribution because an amazing amount of genetic diversity was rescued and saved from extinction. And it was conserved. But there is a story behind these numbers. And that story, you can begin to uncover it by looking at that purple line at the bottom of this graph. Because as the number of samples in gene banks went up and up and up during the 80s and 90s, that bottom row, that bottom line, nothing much happened. What is that bottom line? That's the number of accessions collected by the Consultative Group on International Agricultural Research, the big international gene banks. So in other words, the huge explosion in the number of samples being stored and conserved in gene banks around the world cannot be explained by new material coming into the gene banks, because very little was coming into the gene banks, as you can see from that bottom line. It can only be explained by unintended duplication, which of course is very costly to maintain. This is a symptom, I would argue, of poor information systems, poor coordination, and a poor communication, and poor cooperation amongst governments and gene banks. Certainly there are poor information systems. There are no global information systems. There weren't any a few years ago. So if you needed a sample from the Polish National Gene Bank, you really needed to speak Polish to access that database online. So obviously, not too many people did. And there was limited distribution of materials. And of course, that's because, in part, because there were poor information systems. So here's a graph that shows you different countries and institutions and how much material they've distributed internationally. The big bar on the left are the institutions of the CGIAR, the big international agricultural research centers. The next one is the United States government. The next one is AVRDC, the World Vegetable Center in Taiwan, and then it tapers off after that. So, but there is one thing that unites all of the gene banks, and it's not just that none of them have a secure multi-year budget. It's that we are all interdependent, and I think nothing shows that better than this table. This table gives you a list of countries that Australia, Canada, US, UK, Ethiopia, Brazil, China, India, a number of crops at the top, and the numbers on the page tell you what percentage of the global collection they actually hold. And I simply invite you to consider the fact that none of the numbers on this table are very big. So, in other words, the biggest, most important gene banks in the world still hold a very small percentage of the number of accessions being conserved. So when I travel to Australia, and I've done that a number of times and spoken with people there, they say, we have a great gene bank here, Horsham. We have a lot of wheat samples. We're in great shape. And I say, yeah, you are. You do have a great gene bank. But in your entire country, you've got 3% of the world's accessions of wheat, and you have a very large wheat industry. So if you're happy pinning the fate of your wheat industry to 3% of the world's collection of wheat, be my guest, but I don't know of any scientists that would be too happy about that. That gives people pause, of course. And this goes to demonstrate one important point, and that is that we are our brother's keeper, and that it is important what happens in the different gene banks, even if they're not our gene bank. Why? Because we are all interdependent, or let's say it the other way, we are all dependent on other people's genetic resources. So this is the situation that I found when I came to work at the Global Crop Diversity Trust at the very beginning of that little organization about 10 years ago. There were about 1,700 gene banks. A few of them were great gene banks, but many of them were what I would call hospices. They were places where seeds were going to die. A few were actually morgues. And there were about 7 million seed samples. And we had only about 1.2 million, maybe 1.5 million of those 7 million were actually unique. The rest were just spread around under different names and accession numbers in gene banks around the world, poorly cataloged and poorly researched. So we had a global gene bank system that did not deserve the word system attached to it. Now, the trust, the Global Crop Diversity Trust, was established to ensure conservation and availability of this diversity in perpetuity. I had worked in this field all of my adult life, but I must admit that on my first day of the job as executive director of the trust, it was that word in perpetuity that really caused me pause. Because I thought I knew what it meant to conserve genetic diversity, but in perpetuity, wow, that's a little bit different challenge. So the question that confronted me that day, that confronted all of us, was what are we going to do? And more to the point, what are we not going to do?